So today I will talk about hemolytic anemia. So it is a condition where there is it's a condition characterized by increased destruction of red blood cells and increased production of red blood cells. So before knowing the topic, let us first know about the normal red blood cell. So normal red blood cells are produced in bone marrow, and its primary function is to transfer oxygen to the tissues. The normal lifespan of the red blood cell is 120 days. After that, the old red blood cells are destroyed by spleen and the liver. So in a healthy individual, there is always a balance between RBC production and destruction. So moving on to the introduction, anemia is a condition characterized by decreased hemoglobin level, which is due to blood loss. Red blood cells are produced in bone marrow, and its normal lifespan is 120 days. So as I've already told that all RBCs are removed by spleen and liver, and there is always a balance between RBC production and destruction, because the bone marrow continuously produces new RBCs to replace the old ones which is destroyed by spleen and the liver, which keeps the RBC count and oxygen transport normal in healthy person.
Yeah, the last statement is important. When RBC destruction increase more than production, it leads to hemolytic anemia. Very good, excellent.
So this picture.
It's okay, let's issue it laptop. Or you can discuss it, so we can understand. You will discuss about the diagram, right? Yes. Oh, I mean you will discuss the previous diagram, right?
Yeah.
Difference between anemia and hemolytic anemia. Or you can discuss directly. It's okay.
I was trying to show the picture of a normal red blood cell and hemolytic condition. So, like normal RBC are normal in shape, having bicuspid shape, and hemolytic anemia, the membrane of the red blood cell is usually defective or fragile. It is fragmented or irregular. So, moving on to the classification of hemolytic anemia, we have two types. It is inherited and acquired. Actually, it is based on I don't want to discuss about it, that it's coming now.
It's okay.
So, hemolytic anemia is basically classified based on cause and based on site of hemolysis. So, it is based on cause. We have inherited and acquired types. So, inherited have three types: membrane defects, enzyme defects, and hemoglobinopathy. Acquired involves immune-mediated and non-immune-mediated. So, based on cause, inherited, also called as congenital hemolytic anemia, are due to genetic defect that makes red blood cells abnormal or fragile. So, if we talk about the membrane defect, in this type, the problem is in the outer covering of or membrane of red blood cells. So, what actually happens here is RBC membrane are made of special protein like spectrin and carine. This protein maintains the normal biconcave shape of RBC, and because of this shape, RBC are flexible and can easily pass through some blood vessels and the spleen. So example of membrane defects are hereditary spherocytosis, in which RBC becomes spherical, and hereditary erythrocytosis, in which RBCs become oval or elongated. So we have another type, is enzyme defect. It is an inherited condition in which RBCs lack essential metabolic enzyme, leading to energy deficiency or oxidative damage and premature destruction of red blood cells. Example is D6PD and pyruvate kinase deficiency. So the third one is hemoglobinopathies, which is also called as normal hemoglobin. So it is an inherited disorder caused by genetic defects in hemoglobin. Hemoglobin is the protein inside red blood cells that carries oxygen. If its structure or production is abnormal, RBC becomes weak and are destroyed early, leading to hemolytic anemia. Examples involve sickle cell anemia and thalassemia. Another type of hemolytic anemia involves acquired hemolytic anemia, which is developed after birth due to external or immune causes. So it is also classified into two types, immune-mediated and non-immune-mediated. In immune-mediated, the body produces antibodies against its own RBC. It has two types, autoimmune hemolytic anemia and alloimmune hemolytic anemia.
See, you got.
So in autoimmune, so in autoimmune hemolytic anemia, in this condition, the body mistakenly attacks its own RBCs. In case of alloimmune hemolytic anemia, the antibodies are formed against red blood cells from another person.
Okay, very good. One second, sorry to disturb. Prena, can you explain me what is the difference between autoimmune and alloimmune? What is alloimmune means?
Sir, in alloimmune, it is.
Okay, see students, don't get confused. Yeah?
The antibodies are formed against RBCs from another person.
Yeah, see, if the antibodies came from the same species, not same, different animals. If this is human and another human, one human antibody is attacking another human antibodies, system, that is called alloimmune. Okay, autoimmune means our own antibodies attacking ourselves. Okay, my antibody attacking my cell is autoimmune. Allo means same species. In this case, mother's antibodies will be attack the fetus, developing fetus. That's why it is called alloimmune hemolytic anemia. Classical example is HDM. Very good. And we know autoimmune, we have warm type and cold type, and we know the type of antibodies involved. Warm type IgG and cold type IgM. Can you tell which one is pentavalent and which one is monovalent? Which one is pentavalent among those two? These questions you will encounter in Viva also. IgM. Okay, IgM has 5 antibodies. It's like a ring. Okay, it's like a snowflake. And another one is, what is the first formed antibody in the body? Do you know that? Which class of antibody is the first generated in the body? There are 5 classes, right? GMD, IgM is the first formed antibody. Okay, keep in mind. In Viva, if they ask, you should be able to answer it. IgM is the first formed antibody. And keep in mind, if we identify IgG, that indicates protection against the pathogen. IgG is the last formed and lifelong protecting antibodies. Okay, this is something relevant to immune system, but still keep in mind. Yes, proceed, Miss Dr. Prena. Okay, no, don't worry.
So, sorry, I've already said about autoimmune and its type. There about the autoimmune. I want to discuss more about it.
Yes.
So, autoimmune hemolytic anemia is basically caused due to mismatched blood transfusion.
Very good.
If a person receives incompatible blood, and another condition is SDM, hemolytic disease of the newborn. So in this case, the mother
Okay, mother will attack the baby, fetus. Okay.
mother in this case, like in the first pregnancy, if the baby is Rh positive and mother is Rh negative, during pregnancy some fetal blood enters maternal circulation, and mother immune system sees Rh positive cells as foreign and starts producing anti-Rh antibodies. And in the first pregnancy, antibodies production is slow. Baby is born normal. Fetal and maternal blood are mostly separate. So first baby is usually unaffected. So this is also called a sensitization phase.
And the second baby will get
The
affected.
second baby, no, the mother already has anti-Rh antibodies, and if second baby is also Rh positive, maternal IgG antibodies crosses placenta. They enter fetal circulation. They bind to fetal Rh positive RBCs. So fetal RBCs are destroyed, which causes fetal anemia and severe other cases like hydrocephalus, which is a condition characterized by the accumulation of fluid in pleural cavity and pericardial cavity of the fetus.
Yes, keep in mind now, when Prerna says mother antibodies attacking fetus, keep in mind only IgG can pass the placenta. No other antibodies can pass the placental barrier. Only IgG can pass, and unfortunately in HDM it is IgG class of antibodies that are attacking. If it is other antibodies, it's okay. IgG has tendency to pass through the this one placental barrier. Very good, Prerna. Next.
So talking about non-hemolytic anemia, it is a type of hemolytic anemia where red blood cells are destroyed due to physical, mechanical, and chemical damage, not because of antibodies or immune reactions. In this condition, RBC gets injured or broken while circulating in the blood, which leads to early destruction and causes anemia. So examples are microangiopathic hemolytic anemia. Examples include disseminated intravascular coagulation and thrombocytopenia purpura. Another example includes paroxysmal nocturnal hemoglobinuria, drug-induced mechanical heart valves infections, example malaria.
So, what is paroxysmal nocturnal hemoglobinuria, students? Can anyone tell? What is it? Paroxysmal nocturnal hemoglobinuria. What does it mean? Anyone? Yes, Tessin lab? Yes? What is it? Are you Tessin lab? Yes, Tessin lab. You forgot? Or you want Prerna to answer? Yes, she wants you to be more confident, you know. She wants your charismatic explanation. Yes, Prerna, what is it?
In this condition, RBC have membrane defect.
Urine, no, blood in the urine at night. Paroxysmal nocturnal hemoglobinuria, okay? So, yeah, I hope you know that concept where there are cold antibodies during night, temperature will drop down, cold antibodies will cause hemolysis leading to discharge in the urine in the morning, okay? And I still want to study, actually I'm not getting time. What is the thrombotic thrombocytopenic purpura? Prerna? It's like Prerna purpura, almost similar value. Yes, TTP. What is TTP? Have you went through it? Short definition. Next time every one of us we should know what is TTP, okay? Maybe I will ask that as a 2-3 more questions. What is TTP? Thrombotic thrombocytopenic purpura. Everyone try to understand because that will fall under what is this classification? Non-hemolytic anemia, which is caused due to mechanical stress, you said. Microangiopathic and TTP. Yes, continue now.
Talking about based on site of hemolysis, we have two types: intravascular hemolysis and intravascular.
Okay, bonus point, intra and extravascular hemolysis. It is a 10-mark question for you in upcoming exam. So prepare for it. Yes, Prerna.
In intravascular hemolysis, RBCs are destroyed inside the blood vessels. When the cell breaks, hemoglobin is released directly into the blood plasma. This may lead to hemoglobin in blood, which is called hemoglobinuria, and hemoglobin in urine is called hemoglobinuria. Another type is extravascular hemolysis. So in extravascular hemolysis, RBCs are destroyed outside blood vessels by macrophages, mainly in the spleen and liver. This process often leads to enlargement of the spleen, which is called splenomegaly, and jaundice due to increased bilirubin from RBC destruction. So hemolysis can occur inside blood vessels, intravascular, or in organs like the spleen and liver, extravascular, depending on where red blood cell destruction takes place.
Very good. Now tell me, Prerna, what type of bilirubin will increase in case of prehepatic hemolysis? Before liver, if red blood cells destroyed in the circulation, in the blood vessel, what type of bilirubin will increase? Is it a conjugated bilirubin or unconjugated bilirubin? What bilirubin levels will increase in prehepatic hemolysis? Conjugated. Yes, students. In prehepatic hemolysis, what type of bilirubin will increase? Conjugated or unconjugated? Unconjugated, okay? From today onwards, remember, conjugation happens due to liver only. Liver will make that glucuronic acid. That will make it conjugated, okay? Before liver, it is always unconjugated. Okay? Very good, Prerna. Continue. This is enough. Well done, Prerna. Good job. We covered all aspects of hemolytic anemia, right? Well done. Very good. You can stand there. I will take one picture.